Hi, and thank you for calling in to today's webinar. This is part of the SSPC webinar series and is being first broadcast on September the 18th, 2020. The webinar is on the continuous crystallization of active pharmaceutical ingredients, and I will be delivering today's webinar. My name is Stephen Ferguson. I'm currently an assistant professor at the School of Chemical and Bioprocess Engineering in University College Dublin, and I'm also the team lead for manufacturing as part of the SSPC Research Centre. This is the Science Foundation Ireland Research Centre for Pharmaceuticals. Over the course of today's webinar, we'll of course discuss continuous crystallization of active pharmaceutical ingredients. And to this end, we'll first discuss the types of continuous crystallizers that have been used in the scientific literature and in the industry to date in isolating APIs continuously. Furthermore, we'll discuss the dynamics that these different continuous crystallizers offer in comparison to batch isolation or crystallization, which is the current industry standard, and how these additional process trajectories or crystallization dynamics offer the ability to control and also to access a wider range of critical quality attributes for active pharmaceutical ingredients, namely here the mean size, the morphology of the particulates, the polymorphic or crystalline phase that's isolated and its purity, the chemical purity of the API, and finally the chiral purity or the enantiomer of the API that can be isolated as well. So this is the second part of the webinar, which, as outlined in the first part, is partitioned into two pieces for the purposes of enabling a Q&A session in the time slot available for the SSPC webinar series on the 18th of September 2020. This part covers polymorphism and continuous crystallization, and also purification in continuous crystallization. Polymorphism is the ability for a compound, when crystallized, to exist with multiple crystalline phases or multiple crystal structures. So why is this important to control for the crystallization of APIs? Well, those different crystalline phases can have different solubilities in your small intestine, which thus controls the bioavailability of the drug once dosed, for example, as a tablet in an oral solid dosage form. So as a result, we have to be able to control polymorphism in continuous crystallization if we are to use continuous crystallization techniques in the isolation and crystallization of APIs. So as outlined, there had been previous work prior to the recent spurt of development on continuous crystallization technologies for commodity chemicals. However, really none of these studies considered polymorphism, as these very small molecular weight, often inorganic compounds, did not experience significant polymorphism. So this was kind of an open question that had to be figured out through research, and ultimately the answer was yes, it can be controlled. And this is particularly interesting in the case of MSMPRs. So maybe to start, we'll look at a phase diagram again and look at the dynamics of the systems as we did previously for particle size control. So this is a monotropic polymorphic system, actually glutamic acid here, although not labeled. And these are temperature dependent phase diagrams. So as the temperature goes up, the thermodynamic or the maximum stable amount that can be dissolved in solution goes up. Hence the liquid phase concentration that can be dissolved goes up. So monotropic systems like glutamic acid, the phase diagrams or the solubility curves for the two polymorphic forms don't cross over as a function of temperature or as any of the other process variables that may be manipulated in this case. There is also enantiotropic systems whereby there is a flip over in the stability of the two polymorphs, usually as a function of temperature. So what happens in a normal batch crystallization, which also can exhibit polymorphic issues? So if we start off with an undersaturated solution, which will be clear, we then cool it down and we pass through the solubility curve for the stable form, in this case the beta form of glutamic acid. And we continue to cool. There's no nucleation to date. We're still in the Labol region. We also pass through the solubility curve for the alpha form and the metastable form, or the unstable form of glutamic acid in this case. And eventually we either seed or we allow spontaneous primary nucleation to happen. And in this case, and in many cases, this nucleation favors the metastable form. So because there's higher primary nucleation kinetics for the alpha form, we'll see that appear first. Faster primary nucleation kinetics mean a lower induction time for nucleation. We then get secondary nucleation as these particles hit the impeller and each other and generate a population on which we grow alpha, more alpha crystals. So this way we can deplete the mass down to this first solubility curve. And in a batch vessel, we just leave this stir. Eventually, some beta form will nucleate as there is still supersaturation between the beta form and the alpha form solubility curves at this, which is the isolation temperature. And once this beta form nucleates, it will start to secondary nucleate due to the presence of supersaturation here. And this will then produce a population of beta form crystals, which will have some driving force. These will grow, sucking more concentration of the glutamic acid molecules from solution. This will bring the concentration below that for the alpha form, which will then dissolve, replenishing the L-glutamic molecules in solution. And this will occur until there's no alpha form molecules left, and then we will eventually move down to the solubility curve for the beta form, whereby the solution will be at thermodynamic equilibrium, and isolation of a single phase of beta glutamic acid can be isolated. So continuous tubular crystallizers will follow the same dynamics that are seen in batch crystallization, again, except with the progression of the crystallization or the crystallization dynamics taking place over the length of a tubular section rather than over time in a batch vessel. So they're relatively easy to intuit how these would proceed. However, no one had really studied polymorphism in MSMPR type crystallizers. So there's a couple of things that we posited that would be, can you isolate metastable polymorphs? So if we operate only at one point on the phase diagram, as we do with MSMPRs, the solution-mediated phase transformation that can make our metastable forms transform into our stable forms really doesn't exist. So if we stay with in a position that has supersaturation with respect to both the alpha form and the beta form, in this case for glutamic acid, there is no solution-mediated phase transformation possible in the same way that there would be in batch or a continuous tubular crystallization. So if we operate here, the idea is that we will operate in a state that is kinetically controlled. So essentially the secondary nucleation and the growth kinetics for the individual polymorphs will drive how fast they can take material from solution. And in such, there will be a competition between these two forms to see which will dominate within a MSMPR type crystallization. So this was the proposition that was made, and it did turn out to be an accurate way. And really, we want to see, can we isolate metastable forms and stable forms under this environment? And to look at this, started as per the phase diagrams with L-glutamic acid crystallization. This is a common model system used in the study of polymorphism. And as outlined, 25 degrees, if you're going to do an isolation from a cooling crystallization, the metastable alpha form in this monotropic polymorphic system will dominate kinetically. So at 25 degrees, you see if you cool down to 25 degrees and hold, you only isolate API in the alpha form if you take a sample here. But slowly, over time, you do get some primary nucleation of some beta form crystals, which will nucleate, grow, take supersaturation or take concentration of L-glutamic acid from solution, thus dissolving the alpha form and eventually allowing a batch crystallization of only beta form crystals to be isolated. You can see this is very slow. It takes almost 40 hours for the transformation to take place. This can be temperature dependent, so the temperature dependent kinetics of each of the forms may vary. So if you isolate or hold at 45 degrees, you'll see that initially in the nucleation event, which occurs here, there is some beta form from the beginning, and hence the transformation is much faster, on the order of 7 hours compared to 40 to go to complete transformation from a mixed population in this case to a population of crystals that is entirely beta form crystals via solution mediated phase transformation. So we see that the secondary nucleation and growth kinetics for this process is more favorable for the stable monotropic form at higher temperatures, 45 degrees in this case. And we see in the batch crystallization, we only get nucleation of the alpha form and a very slow transformation to the beta form or the stable form, which would typically be sought in API crystallization processes at 25 degrees, which incidentally, as it's a lower isolation temperature, will yield a higher yield as the solubility is lower at this point. So this was investigated in this paper, which outlines for the first time how to control polymorphism in these types of continuous crystallization systems. And we ran the MSMPR experiments at lower temperatures. Only alpha form was isolated in the crystallization runs for MSMPR systems. And this is at a variety of residence times up to 120 minutes. But when the MSMPR crystallization was run at 45 degrees, it was possible to directly isolate the stable beta polymorph of L-glutamic acid. So this was the first demonstration that was possible to isolate both alpha and beta polymorphs, so metastable and stable polymorphs in MSMPR systems. However, this begged a couple of questions. So first of all, were these steady states stable steady states? So we talked already about how you get oscillatory steady states that would go up and down. So are these steady states that are achieved in the crystallizer going to continually and perpetually produce pure beta or pure alpha, which would be desired for use in API crystallization? So to test this, we conducted an experiment whereby batch crystallization was conducted, and this was allowed to progress to transform to produce a population of entirely beta crystals, so the stable form for L-glutamic acid. And the question was, if we have a seed loading that is very high to start off in MSMPR crystallization, that is the stable form, would the stable form persist, or would we go to a stable or a kinetically stable steady state that produces the metastable form? So essentially, is this a self-correcting and stable steady state that allows us to produce a metastable form at scale, which would be a development and potentially useful in a variety of industries? And the answer was yes. So after the batch crystallization, the pumps were started up and crystallization proceeded, and a steady state that was metastable for the stable form of the API was achieved. But once some alpha form was nucleated at this point, the alpha form began to kinetically dominate. So at this temperature, it had more favorable secondary nucleation and growth kinetics. So it was gradually able to go back to the same steady state that was achieved in the previous runs. So this confirmed that yes, it is possible to isolate metastable polymorphs continuously, and also that these are kinetically stable steady states, so they will self-correct even if we put in a huge seed loading above a 100% seed loading relative to the steady state for the crystallization of the stable form. We will revert back to the metastable crystalline form and isolate only alpha again if we run this crystallization at 120 degrees, 120 minutes at 25 degrees. More broadly, in continuous crystallization systems, if you're concerned or you want to verify that your steady state is a kinetically stable steady state that will be suitable for manufacturing, and you want to do this empirically, competitive seeding experiments would be a way to do this. So going to a steady state and seeding with an alternate polymorph to see if that will go on to dominate, or as in this case, at the most extreme case you can do, of a very high above 100% seed loading, and allow the crystallization to the opportunity or enough time to self-correct if it has the tendency to do so back to the original steady state at which you've selected for manufacturing. So this study that I was involved in Alan Morrison and Bernard Trout's research group in MIT developed a lot of the tools for developing continuous polymorphic stirred tank crystallizations. To date, in the slides demonstrated that it's possible, for the first time in this study, to isolate metastable forms in this way at scale, and also the thermodynamically stable form, which in this case was carried out by operation at 45 degrees. However, if we look at per pass yield in a continuous crystallization system, obviously operating at 45 degrees to isolate the stable polymorph will result in a lower yield. With kinetic modeling, population balance modeling, it can be evaluated very quickly. What would the residence time requirement in a single stage crystallization be in order to isolate down to detectable limits only the beta stable form at a 25 degree crystallization isolation temperature? You can see this is over a 900-minute residence time, which is very long and is approaching something very similar to a long batch crystallization. So this would be fairly pointless and not a strategy where you would usually pursue crystallization, particularly for manufacturing purposes. What would typically be done would be higher temperature crystallization, but use a recycle loop to make sure that no yield losses are incurred for operating at these higher temperatures in order to control the form of the API that is derived from the crystallization. Because MSMPR systems control the polymorphic content within the crystallizer at steady state through kinetic competition between the two forms or three forms present or are possible for a polymorphic system, there is no extra considerations really for enantiotropic systems compared to monotropic systems, as it's really the kinetics of the system that controls the output rather than the thermodynamics, as would be the case in batch crystallization. So this was found to be the case, and a follow-up study here shows the phase diagram for para-aminobenzoic acid. So this enantiotropic system has a high temperature stable form, which is an alpha form, and it has a low temperature stable form, which is called the beta form. So previous batch crystallization study demonstrated how difficult it is to produce the beta form of para-aminobenzoic acid through batch crystallization. So the primary nucleation kinetics are very slow for the beta form of para-aminobenzoic acid, and very difficult to isolate as a batch from a batch crystallization. As a result, the commercial form that you get when you order is the alpha form. So one surprising thing about running this system continuously in an MSMPR for the first time is that the beta form dominated and was by far the easier form to isolate in contrast to all batch studies conducted. So this is due to the fact that the beta form, while having very poor primary nucleation kinetics, so to nucleate in batch was difficult, had good secondary nucleation and growth kinetics, and was able to kinetically dominate the alpha form unexpectedly. But the study here posed the question, what if we do want to continue to isolate this alpha form, that's the commercially available form, using continuous crystallization? And also to attach an additional constraint, so 90+% yield, which is fairly typical for very well-designed cooling crystallization, and also no detectable beta form. So this is 95% polymorphic purity in this case, which was the detection limit for X-ray for this polymorphic system. And again, the residence time was capped for a two crystallizer cascade system at 120 minutes. So it was possible to vary the residence time in both and the temperature of both at which they operated at their steady states. And this was done numerically. So this was again done by population balance modeling. And the results of the study showed that there was regions of attainable polymorphism and yield. And there was a segment of the overall landscape in terms of the crystallization dynamics and possible outcomes that did allow above 90% yield and no detectable beta form to be isolated out of the product from the second crystallizer. Again, emphasizing the importance of kinetic modeling in development of continuous crystallization of APIs, as it would have been very, very laborious to do this study purely empirically to find out if there was a possible operating region that can meet the polymorphic specifications for a given system. So following these studies that showed how to design continuous crystallizations to isolate either stable or metastable polymorphs in a way suitable for manufacturing in MSMPR type crystallizers or their cascades, Mike Doherty's group in UC Santa Barbara did the higher level systems engineering work, and for two polymorphic kinetically competing systems in MSMPRs, they were able to reduce down to 3 parameters based on dam color analysis, a prediction of which polymorph, if the kinetics are available for the crystallization of either of them, will occur at steady state in a continuous MSMPR type crystallization system. So taken together, this will essentially allow prediction of the output of MSMPR type continuous crystallizers, which again now provide a way to manufacture both stable and metastable polymorphs in a scalable way. I didn't discuss co-crystallization or the crystallization of salts here. These mechanisms will apply where there's polymorphism in systems with salts and also co-crystals. There will just be an additional design parameter for weaker salts and also co-crystals where the counter-ion or the coformer that's added to the solution, the stoichiometric excess of that will need to be controlled as well. So there's actually an extra design or control parameter to manipulation of co-crystallization or salt systems. Where there are polymorphs, there will be a similar or the same controlling kinetic mechanism at play for MSMPR systems for their crystallization and continuous isolation. So the final thing that we'll discuss in terms of product quality attributes needed for APIs and how continuous crystallization can deliver these will be purification and continuous crystallization, or to demonstrate that continuous crystallization can meet the purity requirements that are so rigorous and required for drugs to be released and used for human consumption. So currently in batch pharmaceutical production, crystallization is the most commonly used purification technique. So it first of all allows for high levels of discrimination between APIs and impurities. It is highly volumetrically efficient. It is relatively inexpensive to conduct. And also, obviously, it simultaneously allows for final API product quality attributes to be controlled during final API isolation. It also allows for intermediate isolation and stockpiling of material, which has a number of benefits in terms of the batch supply chain for pharmaceuticals currently. There is, however, relatively little material in the literature on continuous pharmaceutical crystallization for the purposes of purification, although there are a couple of notable studies. So the batch crystallization methodology is used. A slow batch crystallization conducted over many hours unagitated is quite a powerful purification technique, as mentioned already. However, when you push crystallizations to go faster and faster, so if you conduct them at lower batch times by accessing higher supersaturations, typically or commonly, the purification efficiency of that continuous operation can be driven downwards. So the ability to purge impurities is reduced. In the case of some crystallization systems, if higher supersaturations and lower batch or residence times are used in the case of continuous. As we've seen in continuous operation, we will use, or at least try to use, relatively low residence times in comparison to batch crystallizations, which have to be slow due to their need for a large amount of scale-up in batch operation due to lack of productivity in that mode of production. So one question is there sufficient ability to purge problematic impurities that have to be controlled to almost uniquely low levels in API crystallizations? So to this end, I'll highlight just a couple of studies. Again, another nice study from Professor Alan Morrison and Professor Bernard Trout's research group as part of the Novartis MIT Center for Continuous Crystallization for Continuous Manufacturing. In this case, cyclosporine, a relatively large molecular weight small molecule API with a molecular weight of 1,200 was utilized. The system has a lot of impurities that are structurally very similar. Such impurities are often referred to as lattice substituting impurities, as they can partition into the crystal lattice of the product as the crystallization occurs due to their structural similarities, which creates a relatively low energy barrier or means of or discriminatory ability by the crystallization process. So being able to purge structurally similar lattice substituting impurities is a high bar that continuous crystallization has to clear in order to be able to be utilized for continuous pharma manufacturing. And in the next couple of slides, I'll present a couple of strategies whereby this can be done. In the case of cyclosporine, the batch crystallization, if allowed to proceed for the same amount of time as the equivalent single stage continuous crystallization, so 3-hour residence time in this case, can get to a 74% yield with 95% impurity. So this will be considered a relatively difficult system to purify in batch crystallization. In the case of the MSMPR, so using MSMPR cascades, it was found that the purification efficiency was reasonable, but the yield that could be achieved at this level of purity was relatively low, and this required 3-hour residence time continuous crystallizers, which obviously has a lot of residence time, long startup times, and has a lot of additional things to consider compared to a batch crystallization. The main reason why the yield was so low for both of these is that at low temperatures, this relatively large molecular weight, small molecule API, has very slow growth kinetics. So to get up to the approximately 90% yield that you would try and target for cooling crystallization, this is very difficult. But in batch, you need a long hold time in batch, or you need a lot of vessels with relatively long residence times in multi-stage cascades for MSMPRs. So given that we tend to want to run relatively short residence times for continuous crystallization operations to access the higher productivity that we would seek in a move to continuous manufacturing, but also to be compatible with high productivity flow chemistry steps upstream of a continuous crystallization process, for example. So to deal with this challenging case, or really any case, it's possible to utilize recycle loops. So in this case, you can take a continuous crystallizer that is fed with a feed solution. You withdraw your mother liquor and your solid phase out. You can take out your solid phase using a filter, which is purified at this point, and the mother liquor can be collected. So rather than discarding this mother liquor as in a standard batch crystallization, this can be, in the case of cooling crystallization, can be evaporated, or at least partially evaporated, to bring it back to the feed concentration, or to partially reconcentrate it and fed back in as a recycle stream back into the crystallizer volume. So in this system, Really, we can get to 100% yield. So if we don't purge out any of our mother liquor, we have a zero flow rate to waste in the system with recycle. The only way for material to leave the system is add solid phase product as gas. So some of the solvent can leave as gas. So really, in this case, there's no waste, or everything that can crystallize does crystallize. This inherently has a downside in that if we don't have a purge stream here, any impurities that are non-volatile will crash out and be removed with the solid phase product. And this would very much include the lattice substituting impurities that would partition both into the crystalline product for cyclosporine, but eventually if they reach high enough concentrations, they'll precipitate out and form their own solid phase that would be mostly impurity. So really, in continuous crystallization, we can run our crystallizer at a steady state that mightn't have the highest yield, but we can utilize recycle strategies in order to maximize yield and purity trade-offs. While this does give you better yield and purity trade-off, it does reduce productivity if you use very high recycle rates back into the vessel. So this is something to consider. You have a trade-off between productivity, yield, and purity. And in the case of cyclosporine, for example, this allowed, instead of a three-stage MSMPR cascade, to move to a single-stage cascade, but go up to 91.8% yield while maintaining equivalent purity for this very challenging API impurity system. So just demonstrating that high yields and purities can be achieved in continuous crystallization. So the recycling strategy outlined would be very effective for cooling crystallizations, where concentration by evaporation won't radically affect the composition of the mother liquor. However, we'd like to be able to extend this to anti-solvent and other types of crystallizations, and to this end, organic solvent nanofiltration can be employed instead of evaporation for concentration prior to recycling of mother liquor. So very briefly, organic solvent nanofilters are solvent-compatible semi-permeable filters that are able to retain molecules that have a molecular weight on the order of small molecule pharmaceutical intermediates, ingredients, and products. So in order to demonstrate that the integration of nanofiltration can allow mother liquor recycling techniques to improve yield and purity in continuous API crystallization that rely on anti-solvent or mixed solvent systems to be conducted, an API example crystallization was conducted that was a combined cooling anti-solvent crystallization from a mixed solvent system in order to isolate the API deferrocyrox and purge an impurity for hydrozenobenzoic acid, which results from its synthesis down to very low levels. So this is both a lattice substituting impurity, and it also must be cleared to a maximum below 4 ppm concentration in the final solid phase deferrocyrox product. In many cases, though, this is controlled to a much more rigorous level in the drug itself. So to do this, we take the previous system where we would have an evaporator and a recycle loop set up with solids removal, mother liquor collection and concentration, and add in a membrane module in order to conduct the concentration. So some of the material will permeate the membrane, and this will go to waste. One interesting thing to note, if the membrane is permeable for the impurity and not permeable at all to the API, the system will not need an initial purge stream and will operate effectively with 100% yield and 100% purity. However, there's always at low levels other impurities in there, so there will be a necessity to do some at least limited purging. But the question in conducting this study was, would it be possible to go to much higher levels of purity using membrane recycling techniques? So in the case of this deferrocyrox demonstrating system, the answer was yes. So the results are shown here for batch crystallization, which could achieve a yield of 90% and reach down to 0.32 ppm level concentration in the product of 4-hydroxybenzoic acid. A single stage MSMPR that had a residence time set to be the same as the batch time for this combined cooling anti-solvent crystallization would have a yield of only 70%, but at this reduced yield there was a low level of impurity or a lower level of impurities found. Again, this is right at the limit of detection for the HPLC method that was available for release. So these are effectively equivalent but very high levels of purification. When a recycling strategy with two different membranes was included, it was found that the yield purity trade-off could be greatly improved. So the yield could be pushed up to over 98 and over 99% while maintaining purity in excess of what could be achieved in the batch crystallization. So the graphs just show the attainment of the concentration in all of these streams versus time as the system as a whole goes to steady state. And just to note, the purge stream here in the crystallization without membrane has equivalent concentration at steady state to the other streams as it should. But if we look at the permeate from the membrane, has a much reduced level of API getting out of the system. So modeling studies showed this could actually be pushed over 99%, but this was good enough for any industrial process. If it could be achieved, you would be very happy with this. There was further studies that used addition of complexing agents that did couple with the impurities to allow their preferential removal, even when the membrane wouldn't be able to discriminate between APIs and impurities. So a generalized technique for complexation and removal of problematic impurities was formulated in follow-up studies in the literature to this one. So finally, just to sum up, we have gone through different types of continuous crystallizers. We've spoken primarily about the most common platform used in the pharmaceutical industry for continuous crystallization, MSMPR type crystallizers and their cascades. We talked about how these different continuous crystallizers have different dynamics and how these in turn can be used to control all the critical quality attributes needed in API crystallization. Maybe a small omission, we didn't discuss the subcategory of purification of chiral resolution or removing enantiomers, as most synthesis is conducted asymmetrically in pharmaceutical synthesis. In many cases, this is more comparable to the removal of a standard impurity rather than a full chiral resolution from racemic conditions. However, I've attached a couple of references for both of these, but won't have time to discuss them today. I'd like to briefly as well thank the various research groups I've been involved in, my own current research group, in addition to Brian Glennon's research group, where I did my PhD and who I've collaborated with since returning to UCD as an academic, Professor Morrison on Trout's research group in the Novartis MIT Center for Continuous Crystallization, where some of this work was done, and also to all other collaborators I've worked with throughout the years. So thank you very much for your attention. I'll briefly pop up the extended reference list and take any questions that you may have at this point. So thanks very much.